Thank you, Leonie, for the invitation to come, and thank you, Chantel, for giving me this very cool title of this talk. I didn't come up with that. It's a Chantel thing. So, I just want to give you a context of my experience so that I can tell you some of these stories. And I alluded to this at the keynote speech, but I've been working in plant conservation for a long time. I started out in northern Arizona at the Arboretum at Flagstaff, and there I conducted 5 reintroductions. Flagstaff is high elevation, 7,000 feet. The reintroductions were done at high elevation, dry conditions. And then I moved across the country to Miami, Florida, which is subtropical, lots of rain, and my team was doing reintroductions like gangbusters. They had $500,000 to do a landscape scale reintroduction of a coastal species, and plus, and I kind of went there and went, whoa, I wasn't really quite sure this was working in Arizona. I wonder if really should I be redirecting my team in Miami to do something different? No, I mean the train was really out of the station and we were doing them. And then I moved back across the country, back to the west, which is really where my comfort zone is. And my team at the San Diego Zoo Wildlife Alliance conducted 3 augmentations of 2 species. And most of these reintroductions are still there, but some of them didn't succeed. And I thought that when Chantel asked me to tell you some stories, that she was really asking me to air some dirty laundry. So I'm going to tell you about some mistakes that I made. But remember, and now when I look back at these things, I go, well, of course you should have known that. But remember that at any stage you are in your career, there are going to be things that you don't know, despite the fact that you do a lot of reading, blah, blah, blah. And like what many of you have mentioned, the world, or I guess your realistic examples, Dave, of you get curveballs all the time that you're not expecting, that you didn't think about. So the first example that I've got up here is the Sentry milk vetch, which grows on the rim of Grand Canyon. So this was one of my early experiments, and it actually, I was asked by the botanist of the U.S. Fish and Wildlife Service, which is the regulatory agency of the Endangered Species Act, to come and help her at Grand Canyon, because the plants on the rim were being trampled by hundreds of visitors to Grand Canyon, because there's a really beautiful view right on the other side of the cliff or the platform where these guys live. And she said, I'd really like you to just try some experiments and see what you can do about getting it established. Well, they had set up a fence, and this was my first experience really with collecting seeds for a rare plant. And these little plants, because they were being trampled, produced about 3 seeds that were the size of a mustard seed, really teeny little seeds. And they were being trampled, and so I had something like a total of a little bit less than 300 seeds. And I thought, well, I have grown seeds. I'll just put them out there, and I'll expect that 98% of them will germinate, of course. So I did that. I did, did I say 300? Sorry, 200. About 200 seeds in 1990. We had two actually seedlings emerge from that experiment. One seedling survived, and it was monitored carefully for 4 years, and then it kind of went to the park wanted to take it on. I actually, as an experimenter, ended up learning a lot of things from this. To speak to what Chantel was saying, I started learning how to do demographic modeling. It was my first reintroduction. And the modeling substantiated that it was a really good idea to put a fence around the population. And when I gave this presentation to my Center for Plant Conservation people, and I was talking about it being a failure because only 1% germination occurred, people came up to me, people who had been in conservation longer than I had, came up to me and said, "That's not a failure, Joyce. We get that kind of germination in the field as well. You're not going to get high germination like you do in a laboratory when you get out in the field." And I went, oh. So it was like my peers were telling me what I was thinking was a failure really wasn't a failure, and that if I had just had 10 times the amount of seed, that really I could have established seeds. And what I discovered was that these cracks in the platform was where the seeds germinated. And then when I went back to see them in 2010, it happened that a friend of mine got involved with working for the Park Service to do a complete restoration of the parking lot that was near this population. And the park did this great effort to do more surveys for the plant, lots of, interest from the National Park Service on it. I saw that actually my one little seedling that had made it had babies. And I hadn't been there to visit them in all this time, but 2010, there they are. And some other people said, "Oh, how do you know those are your babies?" And I said, "Because I know where the seedling was." So that's that story. Next story is Arizona willow. This story involves a willow that was known at this time, it's also in the 90s, to occur in the northeastern mountains of Arizona called the White Mountains. And it was being just hammered by herbivores, just to the point where if you don't have an herbivore on it, it'll grow about yay high. But if you have an herbivore on it, you might have only this much of it growing. And you guys, I know, are familiar with this kind of thing. But there are two agencies that are responsible for the herbivores, two different agencies. The U.S. Forest Service manages the grazing allotments that allow cattle onto public land on the national forest. And the Arizona Game and Fish Department handles the permits for culling or shooting the elk. So I had elk and cattle that were eating this plant. But the agencies would argue with one another that, it's not, that little teeth mark on the stem is obviously a rabbit. They'd say it's a rabbit, not their elk or their cows. And I had this young intern who was saying, why don't these guys just do an experiment and they will learn which of these animals is actually doing this herbivory, which is what we did. So we found a place in the White Mountains where we knew there was a grazing allotment. This was with the permission of the Forest Service, and we knew that elk were also present there. We also, at the Arboretum at Flagstaff, had this giant elk exclosure fence on our property that protected our gardens. And my staff, my boss and administration allowed me to fence off an area within this elk-proof fence. And then he happened to know this guy that had a cow. And, so like, hi, I really could use a cow to come over and just see if it will eat these plants. So I planted them at the Arboretum inside of this elk exclosure and also outside of the elk exclosure, and then fenced every single one of them outside of the fence, inside the fence. And then I remove the cages and allow the cattle to, the guy came with the cow in the back of his truck, backed it up, let the cow out into this pasture. It was kind of like, now I think about that and I think that's really amazing that I managed to get this guy to get his cow over here for me. And the elk just were going to be there or not outside of the fence. But we repeated this experiment in the White Mountains. And the problem with the White Mountains part was that where we were given permission to do the experiment, there were only 3 individual plants, and I don't know whether you can even see them. They are the stems that are hanging off the side into the creek there. It doesn't really quite look like a willow, I'm sure. And I took cuttings of each of those individuals and actually had a kind of a long discussion with the U.S. Fish and Wildlife Service about this, because you have to get all these agencies together and talk about it. And the Fish and Wildlife Service lady would not let me bring in any plant material from any other population. Despite the fact that we had only 3 individuals there, she said, I do not want to do a genetics experiment. And so, so the consequence was, the results were, guess what? Cows and elk both eat the willow. And yet, another thing I learned was that cows are terrible about running over fencing. And if you don't have the strongest, fencing and reinforced stakes in the ground, they're just going to sit on it or something. I'd come back, for the period where they were supposed to, they were only exposed for X amount of time, and then I'd come back, and sometimes the caging was just terrible. But, so this conservative view of the Fish and Wildlife Service was that we actually have a reintroduced population at this site, but it has very depauperate genetic representation, and it turned out that this happened unbeknownst to me. So one of the recommendations in our guidelines is that you maintain even family lines. So if you got tall, big guy, robust guy and a short, stumpy guy, you put out the same number of guys out there so that in time you got both genotypes represented, but allowing natural selection to make that decision. What I learned was that without really paying very close attention to what was going on with the propagation in the greenhouse, I just wanted plants for my experiment, right? And there was this big mama that was much more easily propagated than the other one. And so what went out was mostly that into the experiment, because I wasn't really thinking about it. It was much more of that genotype than the other. And these guys are sexually dimorphic, so it's either all girls or all boys that ended up mostly in that garden. It's actually not a garden, it's in that population, in that reintroduced population. So I've said that. So the next example that I have for you is one from South Florida. This is endangered crenulate lead plant. It's a shrub again. And the history of this plant is that it's a federally listed species that by the time that I arrived in South Florida had most of its wild populations extirpated by development because they were growing on private land. So our botanic garden had representation of the plants from those different extirpated populations as well as the wild populations. And prior to my arrival, the folks that worked at Fairchild had placed plants into a county park where it had been known historically. So in U.S. language, that would be a technical reintroduction back to a place where it no longer was in existence. And what the park told me was that they were really interested in the survival from the bay inland because they were so worried about hurricane impacts on rare plants. And so in those early years, this was prior to my arrival at Fairchild, there was a lot of mortality. And I think again, the team kind of thought, oh, high mortality, it's not a success. They kind of knew it was still there, but they thought, it's really not working too well. Now this is a long-lived perennial, by the way, we at Fairchild were monitoring all of the populations of this plant, and one day my staff said that they saw recruitment. And we weren't seeing recruitment out in the wild population. I said, we're seeing recruitment at this reintroduced population. So I got very excited about it. But it was a little bit like what you were talking about earlier about if you're kind of not that next generation, you're worried about what you put out there, but the next generation, And I said, no, we really have to worry about this next generation. Let's go look at them. And this population still is there. But what I wanted to point out to you was some of the things that we found out looking at the patterns of recruitment in the site. So the black dots, these are two plots where plants were installed, and the black dots are adults that were alive in 2007. So that's approximately 10 years, 12 years after they were installed. And the gray dots are adults that were dead after 12 years. And then the red dots are recruits that came out in 2015. And what you'll notice is that there's a real difference in where recruitment happened, first of all. So one of the plots, there's much less recruitment than another plot. But the other thing that's kind of weird is that in that plot one, where the recruitment is showing up is near a lot of dead plants. And so What this got me thinking about as a lesson to share with you all is a little bit of the concept of hyperdispersal, I guess. And also about seed banks. I mean, I don't know whether these seeds were dumped when these adults were alive and they were hanging out in the seed bank for 20 years before they emerged, but these are baby seedlings that we're detecting in 2015. And so as someone who's going out to check on the status of your population, if you only looked under the live plants, you would have missed that recruitment had happened in this plot. And so there is this, like, seedlings are a pain, yes, to track, but it's important to keep on doing it, and they might show up outside of the plot, I mean, even outside of the original plot that you indicated. So we did look at some patterns of where this recruitment occurred, so we know that they tended to be present in more sandy soils. Soil moisture didn't seem to have as great a factor on them. And just to give you an update, I called my former staff members at Fairchild and said, well, hey, how is that reintroduction doing in 2022? And they had another prescribed fire that went through, which is a great thing for seedling recruitment. So in 2021 they had lots of seedlings show up again, and then in 2022 they had mortality of those seedlings, but still persisting and has a nice even or not even, but many stages are represented in the population. So monitoring over long term is part of the lesson. High mortality in the early years can lead to a false perception that your reintroduction is a failure, and yet this is an example of a population that's persisting for 28 years. It's got recruitment, it's hanging in there. We learned that fire is important to the habitat. After fire, the population flourished. And the other thing is it'd be really great to publish your results so that other people can learn from you. So I did note that in what I said earlier is that our recruitment happened under live and dead trees. Microsite matters. Tracking seedlings is time intensive, but I think that they can reveal a lot about the ecology of your target species. One of the things that I recommended to my staff that began to just get weary of tagging every single seedling was to try doing quadrats of seedlings, as if you got an entire cohort of 50 seedlings and they're all within a 20 centimeter grid. Try doing that, and then next year if you come back and you got the 3-inch seedling or the 3 centimeter seedling versus the 1 centimeter seedling, you can just tag that one and assume that it's a year old. And then you might have to look outside of your original plots is the other idea. So thanks so much, and you all look really tired. Thanks so much to Chantel and to Leonie for putting this together, and congratulations.